Okay, we'll start our session. Our next and final speaker in the GIST track is Dr. Jean-Yves Blay, a very experienced and well-respected specialist in sarcomas and also, well, internationally recognized and active. And also, I think you play a leading role in the European networking of rare cancer centers. So we are very happy that you are here to talk to us about GIST. So please, the floor is yours.
Thank you. Thank you very much. And thank you very much for the invitation. And even more importantly, thank you very much for your flexibility. Thanks to John and to Winan who could take over. Well, I'm very happy to be here with you today because I think there are a lot of new things in the field of gastrointestinal stromal tumors, in particular for medical treatment. So what I would like to do in the coming minutes is actually to discuss the state of the art, but also the things which are probably going to change the landscape in the coming years, or at least address some questions which have been pending in the last decades on the duration of treatment, the nature of treatment, and probably also on new treatments to be proposed. So these are my disclosures, both as a researcher and as a director of the hospital for the different contracts. Starting with molecular analysis, I think I'm speaking to an audience which is quite convinced that molecular analysis is crucial for the diagnosis of GIST, but it is not the only important parameter. We now have a better description of the clinical characteristics of GIST patient, and as a series increase in what is published, we see more and more that these are different tumors, but also tumors with different natural histories across the spectrum of ages, and that with the definition of GIST that we have, we should probably be in a situation in the future to have different guidelines depending on the nature of the clinical presentation, the site of the GIST, the gender, the age, the molecular characteristics. And this is even quite obvious. I'm not a pathologist, as you know. This is actually quite obvious if you look at the histological and immunophenotypal characteristics of the different gastrointestinal stromal tumors. Probably nothing very much in common between the SDH mutated GIST, NF1 GIST, the so-called wild type GIST, and the GIST affected with mutations on different codons, exons of KIT and PDGF receptor alpha. So this diversity is captured by the molecular analysis. And I think it's quite fair to say that even though not everyone around the globe, around the countries, have access to molecular characteristics, probably the best treatment decision that we have once we need to start a medical treatment, and probably if we need to have treatment decision in the field of advanced GIST, it's important to have the driver molecular alteration, because we are dealing here with an heterogeneous, very heterogeneous group of tumor gathered under the same acronym of gastrointestinal stromal tumors, and not all are responding exactly to the same treatment. And interestingly, and I think actually that's quite unique in the world of oncology, this correlation between the anatomical description of the GIST and the presence or absence of specific molecular alteration in different organs, that is puzzling on how do we interpret that in terms of mechanism by which the tumor appear and progress. But this is something that we observe, and we probably, on the basis of this breadth of information published and gathered in the last 20 years, are now able to propose numbers for the epidemiological description of all the different molecular subtypes of GIST. This is probably still work in progress. What is very important, of course, Netherlands and France are very close, but what is very striking is that the incidence, overall incidence of GIST doesn't seem to be very different from different countries around the world. And there are comparative studies from Asia in particular, which seem to bring very similar numbers, probably with subtle variation, but quite similar numbers. Nevertheless, these different tumors have different natural history, have different sensitivity to treatment, and many of these subgroup of GIST have unknown responses to many of the agents that we are going to discuss in a few minutes. And that's probably a field where research should be undertaken. I have in mind the question of real-world evidence, gathering the information from electronic patient records all around the world in networks such as the different national sarcoma networks, but also European reference networks. It's going to be very important to get a better level of information on the rare molecular subtypes of GIST, because this is how we are going to do it better. We already know that the impact of treatment varies according to the nature of the mutation on KIT. We also know that by gathering all the information which is published, we can define response rate for even rare molecular subset of GIST. But still, you can find here, you can observe here that the number of patients is sometimes very limited, and we need more information on this question. I think that's probably one of the next frontier that we have to address. All right. Of course, the definition of the disease, characterization of the disease, molecular characterization of the disease is crucial if you want to have a proper diagnosis and proper treatment decision. And treatment decisions are based on guidelines. In Europe, we work on the ESMO guidelines, but basically the ESMO, the NCCN guidelines, Japanese guidelines are very similar in what are the decision process. And let's start maybe with localized GIST on the question of medical treatment. I think this has been covered previously by Winan, but we all know also that the neoadjuvant treatment being proposed for patients with locally advanced GIST, the adjuvant treatment is now proposed on for a duration of 3 years for patients with high-risk GIST, and we have recently read the latest update of the crucial study comparing 1 year versus 3 years of imatinib, demonstrating very convincingly that 3 years is superior to 1 year, and that is the latest data set on the randomized study. We know that there is a non-randomized study exploring a longer duration of imatinib with 5 years, the famous PERSIST study, which also provides some interesting information on duration of longer treatment. What is high-risk GIST? Well, the definition varies across countries, across research group, but I think that basically the AFib criteria, the modified NIH classification, the contour maps and nomograms are usually what is best used for the decision making process. The presence of mutation in a given hotspot, such as codon 557, 558 of exon 11 of GIST, it could be also something of importance. And as we are going to see in a couple of slides, the chromosomal instability could be also important. So how can we do better? There are 2 studies ongoing, which have started some years ago, which have attempted for high-risk GIST to propose a longer duration of treatment with adjuvant imatinib, with the observation that actually a substantial proportion of patients were actually relapsing after the 3 years. And the question was, can we do better? Can we propose a longer treatment, which will lead to lower relapse rates and hopefully better survival as compared to the classical 3-year treatment. So the SSG22 is ongoing, as is the IMAGIS study. SSG22 adds 2 additional years after 3 years of treatment. IMAGIS adds 3 additional years after 3 years of treatment. SSG22 and IMAGIS have both had some relatively slow accrual, but things are going now better. For the IMAGIS study, we are reaching the end of the accrual with less than 10 patients needing to be accrued. So probably in 1 year from now, I will be able to present the first result of the IMAGIS studies. And this is, of course, these are of course very good news. Of course, still the data are still not known, but we still have some interesting information for the patients who are followed in the study, with very little, actually no relapses in the patients treated for a longer period of time. What about the intermediate risk patients? That's something of importance. We knew from 2 retrospective studies on limited data set that patients with intermediate risk GIST with high chromosomal instability index, SYNSARC index, had a worse outcome with an increased risk of relapse as compared to those intermediate patients, intermediate risk patients with GIST without chromosomal instability since ARC-LO. And actually on the basis of that, our colleagues from Marseille in France proposed a randomized study called GI-GIST, randomizing for intermediate risk patients on the basis of a CGHR reanalysis of the tumor, either if genomic index low, surveillance without treatment. If the genomic index was high, randomization of 3 years versus surveillance versus 3 years of imatinib. The study again has difficulty to accrue, but is progressing nicely. And hopefully, if positive, this could be one of the question, one of the answer to the question which is unsolved, which is should we treat and whom should we treat for intermediate risk gastrointestinal stromal tumor. But as I mentioned, data also comes from real-world evidence. And our colleagues from Italy, Bruno Vincenzi and collaborators, have very nicely put up a retrospective study on the outcome of patients treated with adjuvant imatinib in those patients with whose tumor is harboring KIT exon 9 mutation. Should we give 800? Should we give 400? Nobody knows, no randomized study. This retrospective population of patients probably tells us that 800 has limited added value as compared to 400, while associated with superior side effect profile as well, inferior side effect profile with more side effects, as we all know. So probably another level of evidence which tells us that we need to treat these patients according to real world evidence as well, because I think this informs us for the routine treatment of the patient. What about advanced stage patients? Well, the updates tell us that even in the very, very early studies conducted in the beginning of the 2000s, some patients are still without progression some 20 years afterwards. That's of course very good news, but we still need to do better. What is the strategy to do better? Rotation of the treatment with regorafenib was probably not a good idea. Will it be a good idea to address the same question for the same patient population in using ripretinib or even avapritinib? That's an open question, but at least for regorafenib, no superiority of rotation as compared to the standard continuous imatinib treatment. So what about this classical second and third line agent? What about the new agent? I know that John has presented the topic of ripretinib and avapritinib a few minutes ago. Actually, there are at least five new drugs which are ongoing and have demonstrated activity in this group of disease of patients progressing after a third line of treatment. Of course, ripretinib with Invictus study, and you have seen several times the impact on progression-free survival. What is actually very fascinating for me and probably quite a lesson is the impact on overall survival, which is actually increasing as the follow-up increases, and that's probably a sign that this new mode of action, which is blocking all mutation, has probably a very significant impact in the long term. Another very important information, I think, for patients with advanced GIST who are progressing under ripretinib at the classical dose is that it's probably worth considering doubling the dose on the basis of the intra-patient dose escalation strategy, which was feasible in the Invictus study. We see that some patients can achieve prolonged progression-free survival by doubling the dose of ripretinib, BID as compared to QD. And the swimmer plot in yellow, BID in blue, QD of this patient population is quite informative. Graphically, we see that the duration in yellow is at least superior, sometimes equivalent, sometimes shorter, but anyway, significantly superior to what we have with the blue duration of treatment. In other words, doubling the dose of ripretinib is possibly one of the best treatment we can offer to patients after treated with a single classical dose of ripretinib. Of course, the INTREX study, which was reported by Sebastian at the beginning of the year, which was a randomized one-to-one ripretinib versus sunitinib study in second line, failed to demonstrate superiority for its primary endpoint. Nevertheless, this is probably not the end of the story. We have seen that patients need to be characterized molecularly for that tumor, and probably molecular analysis, subgroup analysis are going to inform us on what is the best treatment for this second-line patient. I'm quite convinced that sunitinib is superior to for some patients, for some tumor types, for some molecular types, and conversely that ripretinib may be superior for some patients and some molecular types, but we don't know yet who they are. And what we know, however, is that the impact on response and impact on quality of life of ripretinib in second line is quite interesting and actually worth exploring further. True also for avapritinib, a remarkable agent with a selective activity on D842V, as John has shown you, with a signal of activity which is unprecedented for this group of disease, actually, who are reaching the results observed with imatinib. But this is not the only utility probably of avapritinib, and I'm still impressed by the activity for other groups of gastrointestinal stromal tumors equipped with other mutations. We have here an agent which is quite active, of course, with some toxicity questions which need to be carefully addressed, but at least we have here quite an active agent, even though not approved for this indication around the world. That's certainly food for thought for future clinical study. Again, avapritinib was tested in third line versus regorafenib. The study was negative, did not reach its primary endpoint. But again, I would say the same comment that for the INTRIGUE study, maybe looking at a group overall is not the appropriate thing to do. And this study, while negative, should inform us on who are the subgroups which could derive more benefit from regorafenib and subgroups which could derive more benefit from ripretinib. That's really work in progress. Third new drug, cabozantinib, nicely reported by Patrick Chowsky and collaborators from the EORTC, showing progression-free survival and overall survival in third-line setting, which was quite encouraging, and activity in all molecular subtypes. This is actually a reminder of the pazopanib study, which was conducted some years ago by our groups, which was a positive study with limited activities of pazopanib over no treatment. But we have to remember that actually this was possibly related to the fact that pazopanib has limited activity in gastric gist, and this conducted us actually to understand that pazopanib is not active if you do not have a stomach, or if the if we are not taking IPPs, if we are taking IPPs, because this is impacting the absorption of pazopanib. So probably for pazopanib, again, selected subgroups of patients could be good candidates. We have rare subtypes, TRK inhibitors, and TRK inhibitors which are active not only for sarcoma, not only for GIST, but for a wide variety of different tumors. In sarcoma group, we see that the GIST proportion is very limited, 4 out of these 40 plus patients, but still significant activity. Some are debating whether this should be called a gastrointestinal stromal tumor. And that's probably an important question, but nevertheless, this is still a treatment which is quite active with and providing significant duration of response, progression-free survival, and of course, overall survival. So what is ongoing? In fourth line plus, fifth line plus, we have several examples of treatments ongoing. There are studies which are just been initiated in the second line setting, in addition to sunitinib, with new classes of agents. There are new KIT inhibitors which are currently being evaluated to reach clinical exploration. Just a few pieces of information on studies which are ongoing and accruing quite well. The Advocacy studies is a study, sorry for the slide which is in French, which is randomizing for patients who have exhausted all other options, imatinib on the basis of the right study versus imatinib plus atezolizumab. The study started very recently and is accruing quite nicely. You may remember that atezolizumab showed some sign of activity for some gastrointestinal stromal tumors in phase 1 and phase 2 studies. Work in progress, as is work in progress the LANVA-GIST study comparing lenvatinib versus placebo in patients in fifth line plus, and with immediate crossover for patients who are progressing in the placebo arm. The study is progressing very nicely. Actually, we have reached about half of the accrual with some signs of activity for lenvatinib. So hopefully we will be able to conclude this study in the coming year. So we see that we still have important medical and research update in gastrointestinal stromal tumor, both for medical treatment and for local treatment, which I have not covered. The fact that we have 5 demonstrated new drugs in advanced phase in the recent years definitely show us that there is still a large level of investment of research and academic researchers in the field of gastrointestinal stromal tumors, both in localized phase and in advanced phase. Thank you very much for your attention, and thank you for your patience and flexibility. That was me running in the forest.
Thank you very much for your presentation, which had quite a high density of information, I should say. And high speed, yes. Are there questions in the room? Online? Everybody is quiet.
So what I will do is send you this presentation so you can have more time.
That would be very, yes. Thank you very much. Thank you very much.
I send it to Mitzi.
I had one question. I know that there you mentioned a new study with lenvatinib. Mm-hmm. I don't know which company that is.
Eisai.
Okay, and I think there's another new drug under study, thesis or something like.
Yes, which is testing an agent, a new agent in combination with sunitinib in the second-line setting. Indeed,
okay.
The study has just started. It's open in some countries and has accrued already, but not in all countries.
Okay, so as you mentioned, there's still a lot going on in the GIST field, and I think that's good for patients anyway. So hopefully there will be new possibilities for treatment. Okay, Jane, sorry, I didn't see your hand.
Okay, don't worry.
You switched on, yeah.
Hello. These new studies, are they just taking place in France or are they in other countries as well?
Actually, these are academic studies where we got only the drugs from the companies. So they are not open in centers outside of France. But what we are doing for patients who wish to join the studies, because these are relatively practical studies, this is oral treatment, and if not oral treatment, the treatment is given only in short courses. It's possible with S2 form to reach some centers in France to get included. And this has happened already in several centers, including mine. So
that's from other places in France or from other countries?
From other countries.
Good.
Good. Yeah. It's not that easy because in France the law tells you that you need to have a S2 form for the reimbursement of the treatment which are not related to the study. But for the majority, I have never seen a patient not able to get a S2 form. So I think that should be feasible. And some patients came from different countries in Europe to get access to these studies. Okay, so these are patients who have failed all other lines of therapy? Exactly. That's true for both studies. That's patients who have been treated with imatinib, sunitinib, regorafenib, and ripretinib when available, and who have no other options. That's right. Okay, and is it concentrating on any particular mutational type, or is it open to all? No, we left it open to all because lenvatinib, the data we had were not guiding us towards the specific subtypes. And actually, that was also true for atezolizumab, which is an antibody against PD-L1, as you know, which demonstrated some activity in both KIT, PDGF, and so-called wild-type GIST in the phase 1 and phase 2 studies, mainly in the form of very long progression-free survival. Okay, so in terms of publicizing this to patients, is it okay for us to go ahead and do that? Please do that. Yeah, okay. So in terms of who they contact to progress this, because for example, I've been contacted recently by a patient who lives in America,
but he has an aunt who lives in London. He's originally from somewhere in Asia, and he's desperate to find solutions, and it's the sort of thing that potentially he would be interested in. So just want to know how I point them in the right direction.
What I will do is that I will on top of sending you the slideshow, I will send you the report, the current report of the 2 studies where you have all the names and sites of the investigators in France. So the patient you're mentioning can contact any of these, and if he does not reach somebody, or if he wants, he can contact me directly as usual.
Okay, thank you very much.
Great, thank you.
Sorry,
Estelle, yeah.
Yes. When did these studies started in France? When did they start?
L'Enva started less than 1 year ago.
Okay.
Atezo started 2 months ago. Well, I think there are 4 patients included in the Atezo study.
Okay.
So yeah, we do not have the budget to put in all countries as we wish, because this is only these are studies supported by the company only in the form of free drugs. That's all.
But do they don't have any plan to expand this trial in other countries?
Actually, that's FSG sponsored studies. They only provide the drugs. I think if we have a signal, and I think we will have a signal, they're probably going to jump to the next step, with larger studies. But at this stage, this is where we are.
Okay.
Any further questions? No? If not, very nice if you send us this information and we can distribute it among our members. Thank you very much for being with us, giving your presentation. So this is the end of the session, I think. So thank you very much. Let's applause.
Thank you very much. I just sent the slides to Michi and Estelle.
Yes,
Okay.
very nice. Thank you very much.